Great. Thanks for the invitation to speak to this audience. So I'll present a story from our lab, and we're a group that I think we're on the interface between biophysics and biochemistry and biology. So I'm really excited to share for this more physics-oriented audience. Okay, so all right, so as kind of just some base knowledge, biology is really compartmentalized by structures called membranes. So here I'm showing you electron microscopy images of bacteria that you just heard about, more like R-like cells that you heard about, and an image of what a brain looks like. And in EM, the contrast agents used for imaging really stain lipids. So what you're kind of seeing here are the lipids in these samples, and you can see there are all these lines and compartments and bubbles, and those are all compartmentalized by membranes. So the fascinating thing that's always driven my research is sort of the chemical diversity that we see for different lipid molecules that make up these membranes. So as I said, all cells on Earth use the same basic structure, that of this bilayer membrane I'm showing you on the top right, to compartmentalize themselves. But the chemistry that they can use to make up these membranes changes quite dramatically. And so lipid composition, really the chemistry and the stoichiometry between different lipid species differs really at every biological scale, in our bodies between different tissues and cell types, but within individual cells, it also differs at the organelle and even at the leaflet level, and it can change in response to various factors. What I'm showing you here are some examples of lipid structures that we see in our own bodies. And what I want to get across is that, there are different lipid classes, like we have our phospholipids that are commonly you see in a textbook, and we have different variants like this plasmalogen in the middle that I'll talk about, and sphingolipids and sterols. But much of the diversity we see in lipid composition is really due to very small chemical differences, whether the chains of the phospholipids get a little bit longer, where they have more double bonds, head groups, just adding a few different atoms to a head group. These are the kind of the chemical differences that really define different membranes in our cells. And a big kind of overarching question is why does biology care so much about these very small chemical differences? So that's where really biophysics comes in. And what biophysics tells us is that very small differences in lipid chemistry can actually have very dramatic effects on the properties and even phases that lipids form. So what I'm showing you here on the right is an image from a detector in an experimental setup called small angle X-ray scattering. So in these X-ray scattering experiments, you take a sample of lipids in this case, but you can use it on other biomolecules. You shoot them with high energy X-rays, typically at a synchrotron, and then you look at the scattering pattern, that results from that at a specific angle. And so when you have a lipid suspension, you often see these kind of bullseye patterns of repetitive rings. So what I'm showing you here, this sample is a lipid called phosphatidylcholine. So it's a phospholipid. It's the most abundant phospholipid in our bodies and in most animals. And so phosphatidylcholine loves to form flat membranes. That's why it's so abundant, and that's why it's used so commonly as a model lipid. And how we know it forms flat membranes is through experiments like this. So in the Sachs profile here, you're seeing concentric rings, and we can integrate out radially, and we get this kind of this profile of what we call Bragg peaks. And what you can see is the Bragg peaks for phosphatidylcholine and water have an even spacing, 1, 2, 3, 4. It continues. And that sort of even spacing is indicative of a lamellar phase, in this case, an L alpha for fluid lamellar phase, where the lipids are forming nice sheets. And the reason why phosphatidylcholine forms nice sheets is because of its sort of molecular geometry. So when it's in a membrane or in a lipid aggregate, we say it has kind of the morphology of a cylinder, sort of like this coffee mug here. And that's kind of a simplification, right? Like it's not its actual molecular geometry, but the average orientation of phosphatidylcholine in a membrane looks like that of a cylinder. So the cross-sectional area of the head group is similar to the cross-sectional area of the tails. And by convention, we say it has a spontaneous curvature close to zero. So it's a low curvature lipid. Now, let me contrast that with another lipid. This is phosphatidylethanolamine. This is actually the second most abundant phospholipid in our cells. And chemically, phosphatidylcholine and phosphatidylethanolamine are very, very similar. If you just look at the differences at the head groups, the phosphatidylethanolamine has a primary amine, and the choline has an amine that's methylated 3 times. So actually, some cells can take PE, phosphatidylethanolamine, and convert it to PC just by doing 3 methylation reactions. But if you look at the Sachs profile of PE, it looks very different. The peak spacings are not even 1, 2, 3. And here they're 1, root 3, root 4, or 2, and that continues. And that's because PE in water is not forming a lamellar phase. It's forming a different type of lipid mesophase, in this case called an inverse hexagonal phase or an H2 phase. So in an H2 phase, the lipids actually invert, so the head groups are inside facing each other. The acyl chains stick out, and they form these long tubes, sort of like inverse micelles that are extended, and these tubes pack hexagonally, hence the H, the hexagonal phase. And the reason that PE forms an H2 phase is because of its molecular geometry as we define it. So PE is a sort of a traffic cone-shaped lipid. Its cross-sectional area of its head group is smaller than that of the tails. Why that is not super simple, but just take me at face value. And so by convention, we say it has a negative spontaneous curvature. And so the interesting question is, why I said PE is the second most abundant phospholipid. It doesn't even want to form a lamellar phase. That's the basis of cell membranes. So an interesting biological question is, why do real cell membranes contain so many lipids like PE that are sort of unhappy forming a flat lamellar phase? So there's many kind of models for why that is. If you look at the level of individual proteins in cell membranes, their activity can really be gated by the presence of these sort of non-bilayer high curvature lipids. This is some data from Sarah Keller from quite some time ago, looking at the open state probability of a simple ion channel called alomethicin. And so alomethicin is really gated by the proportion of PE to PC in the membranes that Sarah did this experiment in. When there's more of these non-bilayer lipids, this ion channel opens up, okay? And why proteins can be gated by lipid curvature has been thought about for some time. One name here, Michael Brown, has really done some nice work and nice modeling on this. And so some of the models that come from that is that high curvature lipids are needed for proteins to conform into different orientations that would otherwise thicken or thin the membrane, which is energetically unfavorable. Another important concept that I'll come back to later is that of a lateral stress profile. So the idea is, even within a flat membrane, there are stresses and attractions within it. For example, at the interface between the solvent and the hydrocarbons, you have an attractive force, and that's due to the hydrophobic effect. But there's repulsive forces as well. For example, the head groups, if they're anionic, have a charge repulsion. And importantly, the tails have a repulsion of steric repulsion. And when you have more of these high curvature traffic cone shaped lipids, that tail repulsion gets bigger. Now, of course, all of these forces have to sum up to zero, otherwise the bilayer would blow apart. But the relative contributions of them and their profiles can be very different depending on their composition. And proteins and other lipids in the bilayer feel that lateral stress profile, and they can be gated by it. So that's another way in which these types of lipids are important. Now, commonly when people think of lipid curvature, they're also going to associate that with membrane curvature. And that's true. High curvature lipids can also support exotic sort of membrane morphologies that are important, especially in eukaryotic cells for certain biochemical functions. I'm showing you here the membranes that we see in mitochondria, which is an important organelle for energy production and many other function in eukaryotic cells. And the mitochondria has a sort of flat outer membrane, but then a highly curved inner membrane that features these cristae structures. And that inner membrane features a lot of these non-bilayer high curvature lipids, which we and others have proposed and shown contribute to their unique morphology, which is important for function. The final example I want to show you is an area, a sort of transient topologies where curvature is even higher in membranes. So the image on the left is showing what one of your synapses look like. All of these are vesicles on the top, and these vesicles contain neurotransmitters. And so for neurotransmitter to get released into a synapse to signal to the next neuron in the pathway, they must fuse with the synaptic membrane, which is the plasma membrane, releasing their contents. And anytime you have a biological fusion event, membranes have to undergo kind of a dance where the lipids have to enter into very high curvature conformations. So the top image on the right, I'm showing you a simulation by our collaborator Alex Sode for what one of these fusion pores look like. So you can see this is kind of when the membranes are just kissing each other. We sometimes call this hemifusion. So they haven't fully fused yet. They're kind of in the process of it. In the bottom is a molecular dynamics simulation of what one of these inverse hexagonal H2 phases looks like. And what I want to show you is that these are not the same structures. It's not like there's an H2 phase when membranes fuse, but the topology of lipids in this fusion pore look very much like the topology of lipids in the H2 phase, these high curvature topologies. And so you also need these non-bilayer lipids for these fusion reactions to occur, and those happen in every cell in our body, and in some cells like neurons are especially important. Okay, that's the background of today's talk, but what I really want to get into is kind of a unique story, and it started in an unexpected place. We were not thinking about these biophysical concepts when we started it. We were thinking about really an organismal feature, which is how different animals on Earth adapt to their environments and the role of lipids in that. And so one sort of extreme environment on Earth that's very common is that of the oceans. So here I'm showing you just a plot, I think I pulled this from Twitter, of the depth, of the crust across the equator of the Earth. And so, we make our lives, in these land areas where you have green, but most of the Earth is covered in water, and that water is predominantly the Earth's oceans. And the oceans are very, very deep. So on average depth of the ocean floor is about 4,000 meters. And sometimes you get even deeper, like in deep sea trenches, like the Mariana Trench, where it gets down to about 10,000 meters. Now, as you get deeper in the water column, the environment changes for organisms. It gets colder, at least in the first few hundred meters. So the deep sea is always very, very cold unless you're near a vent or something. But one thing that's unique about the deep sea is the hydrostatic pressure. So really the isotropic force of the water column above you. And so pressure increases by 1 bar or approximately 1 atmosphere every 10 meters you go down in the water column. So when you get down to 4,000 meters, the pressure is 400 bars or 400 times that in this atmosphere. So it's literally crushing. I think it's about the pressure of an elephant standing on a postage stamp or something along those lines. Okay, so how do organisms respond to that? And there's a lot of historical work that has looked about lipid adaptation in oceans and other sort of extreme environments. And a lot of that has been considered through a model, which I won't get into, but it's called homeoviscosity or homeoviscous adaptation. The idea that membranes need to stay fluid in response to cold and high pressure. So we've been looking at this in a kind of unique system, a unique animal system, comparative system. So on the left, I'm showing you an animal. This species is called Bathysauroi, but it's part of a large phylum of animals, or a phylum of animals called ctenophores. So ctenophores are invertebrates. They're gelatinous, but importantly, they're not actually related to jellyfish. So you don't want to call a ctenophore a jellyfish, but you can call it a comb jelly. And it's called a comb jelly because it swims using these ciliated comb rows. So they have very fascinating biology. They're predators, so they're moving around in their environment. They have a nervous system. They're seeking out prey. Some years ago, or 6 years ago, I met this fellow here, Steve Haddock. And Steve Haddock works at the Monterey Bay Aquarium Research Institute, and he studies marine invertebrates, and his favorite phylum are ctenophores. So this animal on the left, this Bathysauroi, this image was taken in its native habitat about 3,500 meters down. Okay, so we can't go down that deep, but Ambari cruises send out these submersibles. This is an ROV shown in the background. This is one of our cruise photos. And these ROVs, go down and they can look at these animals in their native habitats and also collect them and bring them to surface. Okay. So this animal lives at a depth of 3,500 meters, where the pressure is extreme. It's 350 bar, and the temperature is very cold. And we know it's specialized for those conditions because when we bring it up to surface, it's not very happy for long. So this is the same animal brought up to surface. It's brought up in its same seawater, so that water remains cold. So it's not really heating up. But what does change when you bring it up is it becoming depressurized. So the pressure of the deep sea is lost. And if you look carefully, you can see this animal, it's kind of dissolving away. Really, its dermal cell layers are pulling apart. And if you continue this process, you don't see much of the animal very much longer. And this has generally been observed across deep sea systems, deep sea animals, that they're really specialized for their environments. They don't survive at surface. And that's why you never see really very deep animals at aquaria or anything like that. So the Haddock Lab, over many dives, had characterized the distinctive depth distribution of different ctenophore species. So some ctenophores, like this Leucothea, are really shallow specialists that are only found in surface waters. Others can be deep specialists. They're only found really in the bottom of the oceans. Most are more generalists. They're found over a range of different depths, but they're often constrained in one direction or the other. And so we started to ask, were there any molecular features that defined these sort of depth distributions and their specializations? And I'll just mention here, there was an incredible scientist that really led this whole project, Jacob Winikoff, who was working in Steve's lab and in my lab, and now is at Harvard as a NASA postdoctoral fellow. And he really took this project full on and drove it through. So given this is a physics audience, I want to talk about what pressure does to biomolecules and specifically lipids. So here again, these are molecular dynamics simulations, but they match experimental data. This is just a model membrane of phosphatidylcholine. And so on the left, we're seeing one of these lamellar phases that's fluid. And so you can see the acyl chains have a lot of, it's not a movie, but you can kind of surmise that they have a lot of motion. There's a lot of disorder in the core of the bilayer. On the right is that same system when you apply 1000 bar, which is kind of the pressure at the Mariana Trench. And you can see now the acyl chains have changed. They're really rigid. They're all in a sort of trans conformation. And you can see this is kind of more. Okay, so this kind of structure on the right is often called a gel phase or a solid-like phase. And so pressure can induce these gel-like phases, as can cold temperatures. So if you take like some melted butter and you put it in your fridge and it solidifies, it's a very similar kind of transition on a macro scale, as I'm showing here. And pressure can induce that, but so can cold temperatures. Okay. And so why does pressure induce this sort of gel phase? It has to do with the compressibility of the lipids. So what I'm showing you here is kind of a fun, I show this to students to explain these concepts, but on the left, you see a styrofoam cup. And the styrofoam cup, we have some drawings on it because something we do for fun on these cruises is you draw on these cups and you make little images. I drew this one. And then at the end of the day, everyone pools all their cups together and they put them in a mesh bag. And we attach this bag to the side of the ROV that goes down the next morning. Okay, so the ROV goes down, to a few thousand meters of depth. And everyone's styrofoam cups come back later that day in this really compressed form. So you get these little baby cups. So why is that? It's because styrofoam is a highly compressible material. It has all these air pockets. And so when it's exposed to high pressure, it compresses down to a lower volume state. In contrast, this aluminum can, if it goes down to the depth, it comes back unchanged. And that's because aluminum is an incompressible material. It has a really low compressibility. Now, among biomolecules, lipids are the most compressible. Okay, so that's why their volume is going to change when you put them at high pressure. And that explains why they, for example, will form that kind of gel phase I showed you before, because in the gel phase, the lipids are in a lower volume state than they were when all their acyl chains are moving around in this disordered state. But importantly, the compressibility of lipids is also anisotropic. So if you look at a lipid, a phospholipid, its polar head group is like this aluminum can. It has a lower compressibility. It's not changing much with pressure. But the hydrocarbon chains that make up the core of the bilayer are more compressible. Okay, hydrocarbons are more compressible than most polar solutes. And so they're more like this styrofoam cup. And that means that when you pressurize a lipid, you effectively change its molecular shape, or the average shape it has in a system. And a traffic cone shaped lipid like this phosphatidylethanolamin I showed you before that has a negative spontaneous curvature, when you expose it to high pressure, you can reduce that curvature and it will become even cylindrical. It will lose its curvature. This phenomenon was first shown by Saul Gruner's lab about 30 years ago. So here we're just repeating the same experiment they did. And so what I'm showing you now, these are Sachs profiles of that phosphatidylethanolamin, that PE, that cone-shaped lipid, that's forming these H2 phases at low pressure, at atmospheric pressure or 1 bar. But in this experiment, we're taking that PE, and in that Sachs sample chamber, we're squeezing it, we're compressing it, we're applying pressure. And you can see as we do that, it actually undergoes a phase transition from the H2 phase to the lamellar L-alpha phase. Okay, so it's a pressure-induced phase transition. And importantly, this phase transition is much more sensitive relatively to pressure than it is to temperature. So I won't show you all the data, but temperature is a much stronger modulator of those gel-like transitions, the ordering of the acyl chains. But pressure is a stronger modulator of these non-lamellar phase transitions, going from something like a lamellar phase bilayer to an H2 phase. Okay, so what we started doing during the pandemic is we've asked, can we look at these sort of non-lamellar phase transitions, but not in synthetic samples where we buy the lipids from Sigma Aldrich or Avanti, but rather from real biological samples. So here, a student in the lab, Daniel Milstein, is loading one of these samples in the high pressure sample chamber. And all of these experiments were done at the Cornell high energy synchrotron source, CHESS, which has a high pressure beamline, importantly enough. But this sample shown here on the top right is not of synthetic lipids. These are actually lipids extracted from a whole ctenophore. Okay, and so we do a lipid extraction of the whole body. We pull all the lipids and we concentrate them into these really small X-ray capillaries or sample chambers. But we can actually get really nice diffraction patterns from these biological lipids, which are much more complicated in terms of the chemistry of their mixtures, but we can pull out phase properties. So here I'm showing you kind of the key experiment of this study. We're comparing the scattering profiles of lipids from 2 different animals. So the animal in blue, with the profile shown on the left, is a burrowee. So it's a ctenophore that was collected from surface waters. So its native range is near the surface of the ocean. But importantly, this burrowee was collected in the Arctic. So we're looking at an Arctic sample. And we're comparing it with the animal with the profiles in the salmon color on the right, which is an undescribed species that we call chalfie or chalfiella. And so chalfie exists at the bottom of the Pacific Ocean where the pressure is very high, near 400 bar. The reason we're doing this comparison is because both these animals live in very cold waters. So the Arctic burrowee, because it's from the Arctic, its waters are near freezing, and the chalfie, because it's from the bottom of the ocean, its waters are near freezing as well. The difference in environments here are really just the pressures. And so we're trying to remove temperature as a variable. Now, here I'm showing you 3 different scattering profiles taken at 3 different pressures. The top one is at surface pressure. The middle one is sort of a deep sea pressure near the Mariana Trench, actually. And the bottom one is a very, very deep pressure. And so what you see here is that the Arctic sample, the Arctic burrowee, it starts off as a fluid lamellar phase. It's not quite as clean as the pure lipids I showed you earlier, but we can be pretty confident of its phase. And as you pressurize it, not much happens. The peaks do shift to the left and they change their shape. And that's because eventually it will form one of these gel phases at very, very high pressures. But if you look at the sample on the right, the deep sea sample, it looks very different. Its peaks are starting in a different sort of spacing, and it actually begins as an H2 phase. So at 1 bar, even at cold temperature, these lipids are forming a non-lamellar phase. It actually requires a substantial amount of pressure for the lamellar phase to start appearing. And then at very, very high pressures, sort of non-physiologically high pressures, it also forms this gel-like phase, which we call an L beta, much like the Arctic sample. So now I'm showing you that same data and much more in the form of a phase diagram where we have experimental pressure on the y-axis, so starting at surface and going very deep, and temperature on the x-axis starting cold and going warmer. And you see the 3 predominant lipid phases here, the inverse hexagonal phase in the top right, in the salmon color, the fluid lamellar phase in sort of this gray in the middle, and then this gel phase at the bottom. And where the animals actually live in their native environments is shown in these yellow circles. So they both live in cold waters, but at different depths. And what I want to show you is that in their native habitat, both these animals' lipids form predominantly a fluid lamellar phase. But what's different between these phase diagrams is that transition from the lamellar to the inverse hexagonal. That phase boundary moves down in this deep sample. And so the lipids in all of the samples we've looked at are always, for different animals, are in a fluid lamellar state, but they're not so far from a phase transition to a non-lamellar state. They're kind of, lamellar, but a bit unhappy, maybe a bit frustrated. And that's what we think the sort of the Goldilocks zone for cell membranes really has to be. So as biochemists, we're of course interested in sort of the lipids and the lipid chemistry that drove these differences. And so again, I cannot get into all the data we have, but we did really extensive lipidomics, which is analysis of lipid composition by mass spectrometry techniques of ctenophores across the entire phylum. And that included animals from surface waters across a range of temperatures, cold animals from the Arctic, sort of mesophiles from California, and warm water animals collected in Hawaii, as well as a range of depths from surface animals down to 4,000 meters that I showed you before. And the lipids between these animals, the phospholipids specifically, changed quite dramatically. So, for example, here I'm showing you a plot. These are bar graphs where each color represents a different head group of the phospholipid, that polar part of the molecule. And that can change quite a bit between all of these different species. And importantly, we had a lot of biological knowledge about the evolutionary relationships between these species. So we could understand, for example, when a pair of species recently diverged and one specialized for surface waters and one specialized for deep waters. And that also made this comparative analysis very powerful. Okay, so I'm going to get to one of the really interesting things we found, which was a specific class of phospholipids called a plasminal PE or a plasmalogen. And this class of phospholipids was the only and depth correlated phospholipid class. It increased in abundance as animals got deeper. And actually, our deepest animals, more than 50% of their lipids were this one lipid class. So I'll tell you a little bit about it. These plasmalogens or plasminolipids, they differ from regular phospholipids by the chemistry of one of their acyl chain linkages. Instead of an ester linkage, here you have an ether linkage, and there is a double bond right next to it. So we call these alkenyl ethers or vinyl ethers. And these are the lipids that accumulate in deep species, though there are many other lipidomic changes as well, which I don't have time to get into. So of course, we became very interested in the sort of biophysical properties of these ether lipids. These are phase diagrams of synthetic ether lipids compared to their ester homologs. And I'll just breeze through this very quickly. But what I want to say is that these ether lipids really promote these non-lamellar phases like H2, much more so than even the regular PE I showed you at the beginning of the talk, this cone-shaped non-bilayer lipid. These ether PEs, or these plasminal PEs, are even stronger promoters of non-lamellar phases. And why that is actually not fully well established, but the measurements that we have taken shown that actually these ether lipids just have a larger, more negative spontaneous curvature. So here I'm showing you curvature values for a regular PE and a plasminal PE as a function of pressure. Okay, and so pressure is affecting both of these molecules similarly. As you get increasing pressure, the curvature decreases, but there's an offset here. So the plasminal PE is more highly curved. And if you look at the basis of that offset, the difference is about 500 bar, which is sort of the pressure difference between the bottom of the ocean and surface waters. Because we had this PE measurement, this plasminal PE curvature measurement, we could start asking what is sort of the average lipid spontaneous curvature across the lipidome. And so we could incorporate curvatures from all the different phospholipid species, and this is just a weighted average of them. But it turns out that that value, which we call bar C0, is very robustly correlated with depths, much more than actually any specific lipid class. So that's shown here. All right. Again, I'm running out of time, I believe, but I want to mention an important physics collaborator on the project, Ed Lyman's group in Delaware, also carried out really extensive molecular dynamics simulations of specific lipidomes that we identified, so we modeled them. And in MD, it's actually very powerful because you can actually look at the mixture of different lipids, not just one specific, treating them as sort of ideal compounds, but you can really look at their interactions. And in MD, we can make some measurements that we cannot make experimentally. For example, all of these systems, these are two different animals, a surface cold animal and a deep animal at 1 bar and 1,000 bar in the simulation. They look fluid, right? It's not like the pressure is doing anything to them that you can observe, but you can make some quantitative measurements from these simulations. You can look at, for example, are the lipids, changing their the area that they occupy? Are they getting compressed like they do in those gel phases? They do in both systems, but they do similarly, okay? But another thing we can look at is really that lateral stress profile I mentioned earlier. So this is what a real lateral stress profile looks like across these two different systems. And you can see it varies quite dramatically due to the abundance of these high curvature lipids in the deep sample. And Ed's student, Cecily Vargas Arbano, really did some really nice analysis. You can take this lateral stress profile, you can calculate its first moment, and due to theory, we know that first moment is equal to a product of the spontaneous curvature of the lipids. So if you look here, these two different systems, the deep one shown in pink and the surface one shown in green, they have an offset in the first moment of their lateral stress profile. And that offset is about equivalent to 500 bar as well. So roughly their pressure differences. So these data led to a new model for lipid adaptation to environments, and specifically high pressure environments that we term the homeocurvature adaptation model. And the concept here is that all lipid membranes require this sort of Goldilocks mixture of low curvature and high curvature lipids. So if you take a mesophile, like a surface ctenophore or like our membranes, and you pressurize it, you lose these high curvature lipids due to the effective pressure. And so animals living in the deep sea, they need to counteract this by introducing lipid chemistries that support this Goldilocks mixture, even at the high pressure. But of course, those same lipids, when they're depressurized, may no longer form a lamellar phase. So we wanted to test this homeocurvature adaptation model, but doing that in ctenophores is very challenging. They're not really a model system. So we turned to a simple bacteria that we talked about in the previous talk. So bacteria, we can really genetically modify the type of lipids they make, and we can even introduce enzymes and biosynthetic pathways that are not native to those bacteria. So in this experiment, we took E. coli. So E. coli makes a lot of that phosphatidylethanolamine, but doesn't make any of these ether lipids. It doesn't have the metabolism to do it. But other bacteria like this Clostridium do, and they do it in kind of a unique way. But what we could do is we could take the pathway from making these ether lipids in Clostridium, and we could introduce them into E. coli cells. And now E. coli would make these same types of ether lipids that we see in the deep sea ctenophores. And we can test in the lab the E. coli pressure tolerance or changes to it. We grow them in these sort of very heavy titanium steel chambers. They're called Zobel chambers. So you screw the lids, you fill with water, and you hook it up to a high pressure pump, and you pressurize them. So we sometimes call them bomb chambers. So,
Itay, you have maybe a couple more minutes just to make sure we have enough time for the discussion at the end, just to let you know.
yep,
Yeah,
I'm wrapping up.
thank you.
Okay, so when we grow these ether lipid producing E. coli's in these chambers, we actually see that they grow better than the base strain. Actually, they show no ill effects to the high pressure incubation, at least under these experimental conditions, and they fully survive at 500 bar while the base strain is dying off at these pressures. We can then do the opposite sort of perturbation. So in this case, we're taking E. coli and we're making them make this low curvature PC lipids that I talked about earlier. And so again, this is a lipid that E. coli itself doesn't make, but other bacteria can. And so we can engineer E. coli to make these PC lipids. And when we do that, their pressure tolerance goes down. They really actually die off at high pressure. So at least under these experimental conditions and in this system, we see that changing the curvature of the lipids actually imparts or reduces pressure tolerance. Okay, and then the final part of this story is, again, what happens when you have these deep lipidomes that are filled with high curvature lipids. There's an inherent specialization built into this model because pressure really affects all lipids the same way, as I showed you in some of our data. And so the only way to counteract it is by making lipids and incorporating lipids that themselves have an extreme curvature when they're depressurized. And therefore, these membranes may no longer be stable at the low pressures. So that this goes back to the phenomenon I showed you earlier, where these deep animals melt or dissolve at surface. And when we observe their tissues, their cell monolayers undergoing this process, we think we see a non-lamellar phase transition, where actually their lipids are no longer forming lamellar or bilayer structures. They're forming non-lamellar phases like H2. And that could contribute to sort of the inability of these animals to survive depressurization. So that's the conclusion of my talk. Our paper was published this summer, and so the reference is here if you want to read more. But in summary, we think all organisms must balance the curvature of their lipids. We identified a really important driver of that in animals, which are these ether lipids that are very high curvature. And we think this model also introduces an inherent form of specialization where membranes adapted for high pressure are not going to be able to be functional at surface. And so with that, I'll end my talk. This is my lab. I highlighted all the relevant funding sources and collaborators for this project. And again, I want to emphasize the first author of the study, Jacob Winikoff, who I think is going to be really a leader in this topic in the years to come. Okay, and I'm happy to take any questions if we have time.